Hello there, and welcome to this Carbon Academy tutorial on A-level biology. This is part 2 of the nervous system topic, and in this video we will look at the all-or-nothing principle, saltatory conduction, Pacinian corpuscle, and the retina. So let's get started. Here are the aims of this tutorial. We will explain the all-or-nothing principle, describe how an action potential travels in a non-myelinated axon, describe the factors that affect the speed of an action potential, explain what is meant by saltatory conduction, describe the mechanism of the Pacinian corpuscle, and describe the photoreceptors that make up the retina. Okay, let's start with the all-or-nothing principle. This explains that if threshold potential isn't reached, an action potential isn't generated no matter the intensity of the stimulus, as shown by this blue rectangle. But if threshold is reached, the action potential generated will have the same amplitude, that is, the same voltage, regardless of the intensity of the stimulus. Our nervous system codes action potentials in a binary form, 1 or 0, hence why this principle is called all or nothing. However, how can we differentiate between stimuli intensities that do generate action potential? The nervous system solves this issue by increasing the frequency of action potentials, as shown in this diagram. So essentially, high intensities equal high frequency of action potentials. Now we'll move on to how an action potential travels. In non-myelinated axons, they travel as a wave of depolarization. So what do I mean by this? Well, as the action potential travels, sodium ion channels in adjacent regions open, allowing sodium to diffuse into those regions, as shown in this diagram. The wave generated moves away from the membrane that is in the refractory or repolarizing period. If you think about the Mexican wave, the wave is carried away from the people who are moving their hand down after lifting it up. A point to note is that this wave of impulse travels slower than myelinated axons, which we'll look at next. Let's look at the factors that affect the speed of an action potential. The first one is temperature, and it makes sense, right? Increasing temperature improves conduction in axons, as the ions that diffuse in have a higher kinetic energy. The next one is axon diameter, and larger the diameter, the less resistance to ion flow. Think of it like blowing through a straw versus blowing through a wide pipe. Finally, we come to myelination. In the peripheral nervous system, a special type of nerve cell called a Schwann cell makes myelin, which is essentially a fatty substance. It wraps around the axon and insulates action potentials. This prevents them from being lost along the axon. Remember that some axons can be over a meter in length, so a miscommunication will be bad. Furthermore, myelination causes action potentials to jump, quite literally, and this is called saltatory conduction, which we'll look at next. So saltatory conduction occurs due to myelination. The word saltatory comes from the Latin word salire, which means jump. In myelinated axons, the action potentials jump from node to node, which are gaps between Schwann cells. These nodes are called the nodes of Ranvier, as seen in this diagram. You don't need to remember the name, but if you're interested, no one's going to stop you. In non-myelinated axons, however, the action potentials need to travel the whole length, and as seen in this animation on the left, the transmission of impulse is much slower when compared to myelinated axons. I hope this made sense. Now we're going to change gear and look at a special type of receptor called the Pacinian corpuscle. I mentioned this briefly in my previous video. Pacinian corpuscle is a type of a mechanoreceptor that detects pressure and vibration. The way it works is when you apply pressure, stretch-mediated sodium ion channels open as the lamellae deform. This causes sodium ions to diffuse into the membrane and depolarize it. And if enough sodium ions enter to reach the threshold potential, an action potential is generated. And that's pretty much what you need to know about this receptor. Next we'll look at the photoreceptors of the eye. Photoreceptors are found in the retina and consist of rod and cone cells. First, let's look at the characteristics of the rod cell. Many rod cells connect to one bipolar neuron. Don't worry about what it is, just remember the name. And this causes spatial summation. This is where potentials generated by the rod cells connected to one bipolar neuron add up to generate an action potential. This makes rod cells highly sensitive to light. Spatial summation will be discussed in detail in a later video. And since many rod cells connect to one bipolar neuron, the rod cells end up providing low visual acuity, that is, resolution. This is because the neuron can't specify where exactly the stimulus is, as each rod cell will generate a potential relative to their distance with the stimulus. Another thing to note is that rod cells are monochromatic, that is, they detect only black and white color. As you can see from this image, this bipolar neuron is connected to 5 rod cells, and each of these cells generate a potential when they detect a stimulus that gets added up at the synapse between the rod cell and the bipolar neuron. Like I said, we will learn about summation in a later video. Alright, now we'll move on to cone cells. Unlike rod cells, one cone cell is connected to one bipolar neuron. This means no spatial summation occurs. And this also means that the cone cells are less sensitive to light. However, they do have a high visual acuity as there is no convergence, that is, no adding together of action potentials. Cone cells are found as 3 main types: red, blue, and green. The combination of these pigments generate all the colors we perceive in the world. Let's finish this tutorial by looking at the distribution of photoreceptors, a very common exam question. Rod cells are found in high quantities in the periphery of the retina and overall outnumber cone cells. Cone cells are found in high densities in the fovea, which is found slightly lateral to the blind spot. In the blind spot, no photoreceptors are found because this is where the optic nerve exits the eye. So here is a recap of the aims we went through in this tutorial. We went through a lot of information there, and as usual, use these aims as questions to consolidate your learning. Thank you for watching, and I will see you in the next one.